So we're going to look at what's sometimes called genetics. Genetics is the study of inheritance. And if we look at genetics, genetics began about the same time as evolution. You might remember back in 1859, Darwin published a book on evolution that became the foundation for our study of evolution today. And it was only about 6 years later in 1865 that an important paper on genetics was published. There was a big difference between Darwin's book on evolution and the paper on genetics. Darwin's book on evolution had an immediate impact on how biology was studied. The paper on genetics, which was written by an Austrian monk named Gregor Mendel, was completely ignored. And so let's look at some of the ideas about genetics that were common at the time. One idea that was common at the time was something called pangenesis. And pangenesis was the idea that every part of your body makes something called gemmules. And you could think of gemmules like eggs and sperm, except they're made by every part of your body. They're not just made in the ovaries or the testes. And the idea that your eyes pass down gemmules, and then this would affect the eyes of your children. I guess if you think of these gemmules as kind of being the equivalent of genes today, it's not really that bad a problem. But one of the difficulties with pangenesis was pangenesis allowed for the inheritance of acquired characteristics. Now, acquired characteristics are things that aren't controlled by your genes or your DNA, we would say today. Acquired characteristics are things that you pick up during your lifetime. So during your lifetime, if you get very good at playing basketball and football, your kids will be born and they'll be good at basketball and football. Well, you might throw a ball around with them when they're very young, so they might eventually get good, but we don't think that things that you do during your lifetime, that if you read a lot, that your children will be born knowing how to read. Now, if you're still reading a lot after they're born, they might pick up the habit. They might think it's cool the way children imitate their parents and their older siblings. But acquired characteristics are something that we don't truly believe is inherited. There's a few weird exceptions. There's always a few weird exceptions in biology. Preformationism was another idea that was common in the 1800s. And the idea of preformationism is that if you look inside the sperm, keep in mind most biologists were men, if you look inside the sperm, you'll find something called a homunculus. And a homunculus is a miniature person. You could think of this homunculus as being like an astronaut. And basically this homunculus is curled up inside the sperm, or some of the feminists of the time thought it was inside an egg. And when the sperm or the egg hits the right conditions, that little homunculus bursts out, attaches onto the mother's uterus for pregnancy. And this is why offspring resemble their parents. Now, this might sound kind of silly today, but keep in mind at the time, people had just started using microscopes on a regular basis for maybe 30, 40 years. And they were starting to see smaller and smaller things that had the properties of living organisms. So this seemed like a logical extension of what people were seeing under the microscope. And actually, by the time that Mendel's important paper on genetics was published in 1864 and 1865, happened over a 2-year period, preformationism, this idea of this little astronaut inside the sperm cell, was being replaced by blending inheritance. And the easiest way to think about blending inheritance is if you played with your food when you were a little kid like I did. If you had red Kool-Aid and one of your siblings had yellow Kool-Aid and you mixed them together, you got orange. You got a blend of the two colors. And this was the idea of blending inheritance. People recognized that during reproduction, males produced a fluid. During reproduction, females produced fluid. So it seemed logical that if you mix these fluids together, you would get kind of a blending of their characteristics. Of course, there are some problems with this. If you have a male who's 6 feet tall and a female who's 5 feet 6 inches tall, and they reproduce, sometimes they have kids that are taller than either of the parents. Sometimes they have kids that are shorter than either of the parents. So if we're supposed to be blending these fluids from the male and the female, how can we possibly get characteristics that are outside the range of the mother and the father? Either taller or shorter, or, whatever we happen to be talking about. And these were some of the ideas that were common in the mid 1800s. And as I say, they might seem, kind of nonsensical today, but if you view them from a historical perspective, from what people knew about how nature worked, they kind of all make sense. They're things that we have disproven today. Now, keep in mind, when people started seeing chromosomes, it started to change things. The first person who actually identified chromosomes was George Newport, an Englishman, in 1854. Remember, the work on genetics is published in the mid 1860s. So this is only 10 years before then. He found that when he looked at frogs, the sperm, not the fluid part of the semen, was what went into the egg when the sperm fertilized the egg. In 1873, so about 10 years after this period, the first observations of mitosis were made, so cells dividing. So keep in mind, when Mendel proposed his ideas, people didn't even know for sure that chromosomes were the genetic material, or the genes on the chromosomes, if you want to be a little more specific. They didn't know that cells divide by mitosis and meiosis. They knew cells could divide, but nobody had really clearly observed it. And in fact, a lot of biologists thought the proteins on the chromosomes had to be the basis of inheritance. Why? Well, a typical cell contains thousands of different proteins. So think of all the different characteristics that just a person would have: eye color, hair color, skin color. Do they have freckles or not? How tall are they? How big is their foot size? What blood type are they? Are they A, B, or O? Are they A positive or A negative? So there's all these sorts of things that are related to proteins. So since there are so many proteins, a lot of biologists thought proteins were the key. And when we look at DNA, there's a problem. DNA is a really huge molecule. It's millions of nucleotides long. But the problem is there's only 4 different types of nucleotides. Every nucleotide in DNA contains deoxyribose sugar. Every nucleotide in DNA contains a phosphate group. Every nucleotide in DNA contains a nitrogenous base, which just means a base containing the element nitrogen. And here's where we see our 4 differences. There are 4 different bases: A, adenine; C, cytosine; G, guanine; and T, thymine. But there's only 4 different types of bases. This didn't seem like there was enough variation in the DNA to explain all the different characteristics that we see with people. It turns out that if we think about the DNA, one way to think about the DNA is it's like a set of instructions that run a computer. And just like when we look at programming languages in computer science, there aren't necessarily a lot of variations, but it's how we combine all the statements in a programming language. So when we look at Mendel, Mendel didn't talk about genes. He didn't talk about chromosomes. He didn't talk about DNA. All of that was unknown at his time. When he was talking about genetic material, he talked about what he called factors. And he studied inheritance in pea plants in his garden at a monastery. And he studied peas for 2 reasons. First of all, they were an important economic crop. The monastery grew peas and sold them at the market. But also, there was a little bit known about their genetics. So Mendel had a little bit of a head start thinking about how inheritance worked in peas. And he actually started in 1856, and he didn't complete his work, or he didn't go public with his work, maybe I should say, until 10 years later. And when he did go public with his work, it was pretty much ignored until 1900. And in 1900, there were 3 different groups of botanists. Botanists study plants, and they were looking at genetics in plants. And as they were doing their work, these 3 groups who weren't communicating with each other all came to the conclusion that, wait a minute, what we're seeing is what this guy named Mendel described 34 years ago. So that was when he became, in a sense, famous. Too late for him. He had passed away about 10 years earlier. So let's look at Mendel's experiments and what he said about genetics. So Mendel studied 6 different characteristics in his pea plants. He actually started out studying about 14, and as he studied 14, apparently Mendel was pretty smart, maybe too smart for his own good. He kind of knew what he should see, what his results should be before he started his experiments. And as I say, he started out studying 14 characteristics. He ended up throwing 7 of them out because 7 of them didn't behave the way he thought they should behave. They weren't inherited according to what later became known as Mendel's principles or Mendel's laws. So Mendel studied 7 characteristics that were very easy to observe and to tell apart. Now, when we look at characteristics and we think about observing physical characteristics, we'll talk about this some more, but the physical characteristics are something that we call the phenotype. So think of physical characteristics, it starts with pH. Phenotype also starts with pH, not with F. And Mendel picked 7 characteristics where it was pretty easy to tell them apart. Some peas produced round seeds. Notice the seed is just what we would call the pea. It was called the seed back in the mid-1800s. Some pea plants produced wrinkled seeds. Some pea plants produced yellow seeds. Some made green seeds. Some pea plants had purple flowers. Some had white flowers. Some had pods that Mendel called inflated, that were kind of expanded. Some pea plants had constricted pods that were kind of pulled tight around the peas. Some pea plants had yellow pods, some had green pods. Some pea plants made axial flowers. An axis is a main part of something, and axial flowers come off the sides of this main stem. Some pea plants made terminal flowers. Terminal flowers were at the end, at the top of the stem. Some pea plants were tall, and some were dwarf. Mendel actually called them short, but it was later discovered that they had a mutation and they were abnormally short. They were dwarf. So Mendel studied these 7 pairs of characteristics, and he basically did the same experiments with all of them. That's one of the reasons that his work on genetics, when it was rediscovered, was very convincing, because he was very thorough. So let's think about what he did. So let's look first at round peas versus wrinkled peas. And here's a photograph of an actual round pea versus a wrinkled pea. We can see the contrast a little bit better against a black background than a white background, but it's very easy to tell them apart. Now, what Mendel did was he took plants that produced seeds, and when you grew those seeds, you got more plants. He wanted plants that all the plants that came from them gave round seeds. And so he took plants that produced mostly round seeds. He crossed them with other plants that produced mostly round seeds. When we say he crossed them, you could think of another way to say that would be he bred them. until finally he got a line of pea plants that always made round seeds. Now notice this particular photo shows a round yellow seed, but Mendel didn't really care about the color. When he was working on round seeds and wrinkled seeds, all he cared about was whether the seed was round or wrinkled. All the other characteristics were irrelevant as far as he was concerned. So when he got a line of pea plants that always produced round peas, he called them true breeding. They were true in the sense that if you crossed them with another plant that made round peas, all their offspring would make round peas. You never got any wrinkled peas in the family line. Now, he did the same thing with plants that made wrinkled peas. He crossed plants that made mostly wrinkled peas with plants that made mostly wrinkled peas until he had a line of pea plants that always produced wrinkled seeds, never produced round seeds. And again, he called this his true breeding line. So this was the first thing that Mendel did for all his experiments. He did the same thing in terms of producing a true breeding line of plants that made yellow pods and a true breeding line of plants that made green pods. Now what Mendel did was he took a plant from the line that was true breeding for round peas, and he took a plant that was true breeding for the line from the line for wrinkled peas, and he knew these were stable characteristics because they had persisted for several generations, and he crossed them. So a plant from the line that produced round peas, a plant from the line that produced wrinkled peas. And when he did this, all of the peas in the next generation were round. The wrinkled peas completely disappeared. They weren't visible in any of the offspring. None of the offspring produced wrinkled peas. And this was a little bit startling because he knew that this characteristic of wrinkled peas was stable. As I say, he had created true breeding lines that gave wrinkled peas for generation after generation. Now, this type of cross or breeding in genetics is called a monohybrid cross. Mono means one. Hybrid refers to two different characteristics, so whether the peas are round or wrinkled. And a cross just means a mating or a breeding. And so Mendel artificially pollinated plants, and he used pollen from plants that made round seeds and fertilized the eggs from plants that made wrinkled seeds. He used pollen from plants that made wrinkled seeds and fertilized plants that made round seeds. And all the offspring made round seeds. None of them made wrinkled seeds. So this was confusing. A seemingly stable characteristic had disappeared. So he did a second experiment. He took some offspring from this first monohybrid cross. So the first monohybrid cross was taking a plant from the true breeding round pea line and a plant from the true breeding wrinkled pea line. So their offspring are called the F1 generation. F means filial, so they're called brothers. So the first filial generation. So now he took two plants from this and he crossed them. And when he crossed them, this time, even though all the plants had round seeds, he did this and he found out that about 3/4 of the offspring from this cross made round seeds and only about 1/4 made wrinkled peas. And keep in mind, all these experiments that we're talking about, he repeated with each of the 7 characteristics. So he had a characteristic that seemingly had disappeared in the first generation, but then in the second generation, it reemerged. So we'll get to his laws that he developed and kind of how he tried to explain what had happened. So here's a visual representation using flower color. Remember, the pea plants he was working with make white flowers or purple flowers. So the original parental generation is sometimes called capital P. So the first generation, F1, is the offspring of these two plants, and they all made purple flowers on the plants that they produced. Then he took two purple flowers from here and he crossed those, and he got about 3/4 purple and 1/4 white. So he got similar results with his other experiments. In the one we were talking about, he got about 3/4 producing round peas and 1/4 producing wrinkled peas in that F2 generation. And then for his last set of experiments, he did something called dihybrid crosses. Now, di means two. So dihybrid crosses look at two characteristics at the same time. So if we go back to our peas for a minute, he developed a pure breeding line or a true breeding line that always produced round yellow peas. And he developed another true breeding line that always produced wrinkled green peas. So they were true breeding for two different characteristics. So we want to keep that in mind. So this was a dihybrid cross. He was looking at the color of the peas and whether the peas were round or wrinkled simultaneously. So he did this, and when he did his first cross, he got exactly the same results as when he was only looking at one characteristic at a time. So he crossed round yellow seeds with plants that made wrinkled green seeds, and all their offspring made round yellow seeds. Wrinkled seeds disappeared from the next generation, and green seeds disappeared, which was the exact same thing that had happened in Mendel's monohybrid cross. Then he did the same thing. He took two plants that he got from this first generation and crossed them again. And when he crossed them again, since he's looking at two characteristics, this is still a dihybrid cross. But this time, he got some plants that made round yellow seeds, some that made round green seeds, some that made wrinkled yellow seeds, and some that made wrinkled green seeds. So he got all the possible combinations. But when he analyzed it, it turned out that about 3/4 of the plants made round seeds, and about 1/4 of them made wrinkled seeds. And that was the same results he had seen in his monohybrid cross. And about 3/4 of the plants made yellow seeds, and approximately 1/4 of them made green seeds. Again, about the same results that he had seen in his monohybrid crosses. So Mendel analyzed his results, and he developed what are usually called his laws of inheritance. Sometimes they're just called his principles of inheritance, because as we'll see tomorrow, there are actually quite a few exceptions to these laws. Not too surprising, if you think about it. Mendel's work was done about 160 years ago, so he really explained the most common type of inheritance. But it turns out there are a lot of types of inheritance that he missed. We know historically, that's actually why, remember I said he started out studying 14 characteristics and ended up just looking at 8. Some of the exceptions to his laws, that's why those 8 characteristics didn't work. So his first law, he said that if we look at the factors controlling inheritance, they're not fluids. Well, notice this contradicts the basic idea of blending inheritance. So this might be one reason why his work was ignored for 34 years, because he was really going against the common beliefs of the day. What he said about these factors And he basically called them factors. He didn't call them, you know, we might think of them as being the homologous chromosomes or the genes on the homologous chromosomes. He didn't use those terms. He said these factors controlling inheritance are solid particles, and each individual has 2 of these particles. So notice you could think of these as being the 2 alleles that you have on homologous chromosomes. You know, the set of instructions for something like eye color that you inherit from your mother, and the set of instructions for eye color that you inherit from your father. So Mendel didn't use this term. Keep in mind, again, at this time, none of this was known. People hadn't studied meiosis yet. They didn't know that the genetic material was even on the chromosomes, for sure. Now, Mendel also developed what he called his law of segregation. If you segregate things, you separate things. And Mendel said if we look at these 2 factors, they get split apart during reproduction. Well, remember when we talked about meiosis the other day, we said the pair of homologous chromosomes separates from each other in meiosis. And we talked several times about the idea that when you get an egg from your mother, it has 1 copy of chromosome number 1. When you get a sperm from your father, it has 1 copy of chromosome number 1. Even though all your mother's body cells had 2 copies of chromosome 1, all your father's body cells had 2 copies of chromosome 1, you only get 1 from each parent. So this separation makes sense in terms of what happens during meiosis. And then Mendel came up with what he called his law of dominance. He said that one of these factors can always be seen if it's present. So if we think about his peas, in Mendel's peas, round seeds are dominant, wrinkled seeds are recessive. Yellow seeds are dominant, green seeds are recessive. Now, most of the time, it doesn't matter what's dominant and what's recessive, unless your 2 alleles are different. If you're a pea plant and you get 1 allele for round seeds and you get 1 allele for wrinkled seeds, you're going to produce offspring that make round seeds. So when we look at individuals that have 1 dominant allele and 1 recessive allele, the dominant allele is the one that we see when we look at them. So we want to keep that in mind. Now, the other factor, the one that's not dominant, so being wrinkled or being green, if we're looking at the characteristics above, is said to be recessive. And it can be hidden so that it's not visible if the organism has 1 dominant allele and 1 recessive allele. So to show a recessive allele in your phenotype, if you were a pea plant, the only way you could make green peas was if you had 2 copies of the allele for green peas. If you had 1 for green peas and 1 for yellow peas, you would make yellow peas because the yellow color is dominant. Now notice we can't tell which color is dominant and which color is recessive by just guessing. We actually have to do some genetic crosses and see what the results are. It's not as simple as, you know, you can just say, well, obviously red is dominant over green. Not necessarily when we're looking at how the genetics work. So we want to keep that in mind. So we'll work some genetics problems later and we'll talk about what's dominant and recessive. And the reason we know what's dominant and recessive is because there have been some studies done. So the phenotype, as I said, is the physical characteristics. So this is whether we're talking about round or wrinkled seeds, whether we're talking about yellow or green seeds. Now we can't see the genotype The genotype is the actual alleles that the pea plant or the person or whatever type of thing we're talking about has. Now, there's 3 different types of genotype. Some organisms are homozygous. Homo means the same. Zygous refers to the alleles. And organisms are homozygous dominant. I seem to have repeated that twice. Organisms are homozygous dominant if they have 2 dominant alleles, one from each parent. So if we have a pea plant and it got the allele for yellow peas from mom and the allele for yellow peas from dad, that would be homozygous dominant. If we have a pea plant and it got the allele for green peas from dad and the allele for green peas from mom, then that would be homozygous recessive. That plant has 2 of the recessive alleles. So when we say something is homozygous, we can't just say it's homozygous. We have to say that it's homozygous dominant or homozygous recessive. The other possibility is an organism is heterozygous. Hetero always means different. So for something that's heterozygous, it got a dominant allele from one parent. It got a recessive allele from the other parent. So it got the allele for yellow peas from one parent, the allele for green peas from the other parent. Now notice this idea of dominance explains how you could have a characteristic like wrinkled peas that disappears in a generation and reappears later. And the same thing happens with people. I have some friends and both of the parents have brown eyes and they had a child who had blue eyes. Well, guess what? Brown eyes are dominant over blue eyes. So if you get one allele for brown eyes and one allele for blue eyes, you'll have brown eyes. But if you have one allele for brown eyes and one allele for blue eyes, and so does your partner, some of your children should end up having blue eyes, if you have enough children anyway. And that's because, going back to what we said about Mendel, about 3/4 of the offspring have a dominant characteristic like purple flowers. About 1/4 of the offspring will have a recessive characteristic like white flowers. So if you're heterozygous, you could have a child who has a characteristic that's genetically different from you. If you're homozygous dominant, your kids are going to pretty much look like you. So this explains one of the things that we sometimes see at the hospital, which is you have a bunch of people gathered around a new baby and they're saying, man, it really looks like the father or the mother. Why? Well, maybe that parent has a lot of dominant alleles. So when the baby inherits those dominant alleles, they show up in the baby's phenotype. This is what we see when we look at it. Now, from his dihybrid crosses, Mendel developed what he called his law of independent assortment. And basically the law of independent assortment says that the separation of one pair of factors, and remember when we say factors, we're really thinking one set of homologous chromosomes. This doesn't affect the separation of any other pairs of factors. So in other words, if a pea is smooth versus wrinkled, that doesn't really affect whether it's yellow versus green. So this is why in his dihybrid crosses, even though he was thinking about 2 characteristics, he still got about 3/4 yellow and 1/4 green, about 3/4 round and 1/4 wrinkled. So he got basically the same results as he did in his monohybrid crosses. Now, the next thing we're going to do is we're going to work some problems and see if we look at the genotypes of some parents, what can we predict for the range of offspring that they'll produce. but we're not going to do that till tomorrow. So are there any questions about kind of the basic ideas of genetics? We'll work some problems in class tomorrow, and hopefully that will help clear up any questions you have. But if you have any questions today, go ahead and stop and ask them. No? Okay, well, if not, we're going to